Students, now we are about to talk a new topic, that is cloning in yeast. Why cloning in yeast is so much important? Because yeast is the simplest form of eukaryotic organism. We have already studied uh, many cloning experiments in prokaryotic organisms. Thing is that the prokaryotic and eukaryotic systems or cells, they differ drastically. We have already studied that a eukaryotic cell having the cell organelles like a nucleus, endoplasmic reticulum, Golgi complex, mitochondria, chloroplast, etc. These kind of compartmentalizations are not present in a prokaryotic cell. These compartmentalizations are more important because each of these compartments having their own ambiances and their own enzymes and so their own functions. For example, endoplasmic reticulum.
So, do you know what are the functions of endoplasmic reticulum? The most important function of endoplasmic reticulum is that it helps in the proper folding of various kinds of proteins. And another function of the endoplasmic reticulum is that most of the L-linked glycosylations are happening within the endoplasmic reticulum. And then coming to the Golgi complex, most of the oil linked glycosylations are happening in Golgi complexes. So these kinds of compartmentalizations are most important for the proper expression or the proper functioning of these proteins. So these kinds of compartmentalizations are not present in prokaryotic organisms. So expression of these sorts of eukaryotic genes are not possible in prokaryotic organisms. That's why cloning in yeast or yeast as a model organism for cloning the eukaryotic genes are most important. Another most important thing about the yeast is that we can grow them in a cheapest way. That means we can grow them in a simple medium or in a simple lab. It actually cannot be possible with other eukaryotic cells because they need much sophisticated lab setups or a complex medium. All of these things are renders cloning in other eukaryotic cells so much expensive. Other most important things about yeast cells are we are well aware about its genetic map and its genetic markers, selection systems. And these cells are not very sensitive to the common antibiotics. And as we said, the post-transcriptional translational systems are similar to that of eukaryote, and they are single-celled organisms. These features renders yeast as a model organism for cloning eukaryotic genes.
So now about the genetics of a yeast cell or Saccharomyces cerevisiae. They are haploid organisms and it's a genome having a size of around 14,000 kb. That means around 4,600 centimorgan. In the case of yeast cell, 1 centimorgan that comes around 3 kb. Normally this centimorgan that is a measurement in genetic mapping, and kb that's a measurement in physical mapping. Normally this physical mapping is quite accurate, while than that of the genetic mapping. So back to the genome of yeast cell, it comprises of 4 different DNA. They are chromosomal DNA, mitochondrial DNA, 2 plasmid, and in some strains of the yeast cell they have a killer plasmid as well. That means its genome comprises of chromosomal DNA, mitochondrial DNA, and 2 plasmid. And in some strains of the yeast cell they are having killer plasmid DNA too. And coming back to the chromosomal DNA, it encodes around 16 metacentric chromosomes, and altogether that encodes around 312 genes within these chromosomes. So that much about the genetics of yeast cell and about the yeast vectors. There are four different yeast vectors that had been constructed from yeast genome. They are YIP, YRP, YEP and YAC. YIP means yeast integration plasmid, YRP means yeast replicating plasmids, YEP means yeast episomal plasmid, and YAC means yeast artificial chromosomes. Okay,